Great. Welcome, everyone. It's great to see so many of you. We've opened up our colloquium series to campus and community, and it's great to see all of you. I want to welcome you to our second spring talk in our Israel Studies colloquium series, and I'm very pleased to introduce Daniela Kaufer, whose talk is entitled Lessons Learned from Football Players and Epilepsy to Understand Brain Aging. So now to introduce Professor Daniela Kaufer, who is the class of 1943 Memorial Chair Professor of Integrative Biology and Professor of the Helen Wills Neuroscience Institute. She is also the Associate Dean of Biological Sciences at UC Berkeley. Professor Kaufer received her PhD in molecular neuroscience from the Hebrew University of Jerusalem and was a postdoctoral fellow at the Departments of Biological Sciences and Neurosurgery at Stanford University. Professor Kaufer's lab, in collaboration with the lab of Dr. Alon Friedman from Ben Gurion University of the Negev, has discovered one of the biological pathways that lead to age-related cognitive decline. Kaufer's lab studies how the brain deals with perturbations like stress and brain injury and the mechanisms underlying trajectories of resilience and vulnerability throughout life. The research has been published in top journals, and Professor Kaufer is the recipient of numerous awards, and we're very excited to hear from her today. So, Professor Kaufer, I turn the floor to you to begin, and thank you so much for joining us today.
Hi, thanks, Rebecca, and thank you for inviting me. I'm very excited to be here today and share this with you guys, and I'm going to start by sharing the screen. Okay. Can you guys see it? Yep. Great. Oh, and I see that I get the admits now. Okay, I hope I'll see them in time. All right, so very exciting to be here today. I see some familiar faces and some new people. And the idea that there is such a forum is actually something that I'm very excited about. And in my hat as the Associate Dean, I'm trying to do all sorts of programs to encourage scientific collaborations between, right now between Berkeley and the Weizmann Institute, but overall between Berkeley and Israel, and love to see that this is highlighted here. My lab is here, and I want to highlight first the people who actually do the work. These are the current lab members and the alumni, all people that were involved in that project. We're very lucky here, as you know, so fantastic graduate students and undergraduate students even that were involved in this work over the last 15 years or so where I've been at Cal. Usually we're on campus in the Lika Shink Center for biomedical research, but as you can imagine, it's been mostly Zooms in the last 6 months or so. This is a collaboration with a lot of different people, but really highlighting Alon Friedman, who is has a split lab between Ben Gurion University and Dalhousie University, and we've been working on this for many, many years together. And lastly, the most important thing, the one thing that actually enabled us to do this is funding for those projects. And I'm so excited to see that Barbara Baker is today, because you can see here, and I'll get to actually talk about it later, but I was a Baker Fellow with an amazing program that Barbara and Gershon started with the campus, taking sort of ideas, all sorts of ideas that the faculty might have, and translating them into the world. And so we'll get to that later, but really this is what funded this when it was a very wild idea and not something very likely to get funding from anywhere else. So we're going to start with a little bit of a data collection, and there's a lot going on there. So I want to point your attention to some different things that are going on, right? So what we're going to see here, this is the year. Do you see here in the gray background where it says 1800? So we're going to progress in history from 1800s until today. You're also seeing at the x-axis an interesting measure, children per woman, so total fertility. So we're starting in the 1800s, and we're seeing that most of the countries in the world were around 6, so somewhere between 4 and 8, but there's an average of 6. You're seeing here all sorts of different colors, and they represent where they come from in the world. And so if the Americas are going to be yellow and Africa is blue, and so on and so forth. And then on the y-axis is really what we're paying attention to here is the life expectancy in years. So in the 1800s, we're somewhere between 30 to 40 years old as the life expectancy. Okay? And now you're going to follow all of this different information as we're progressing in the years. The orange is Europe. And there's a lot of different things that one can get from this, from seeing all of those different trends. One is a geographical discrepancies, one might say, but a very, very clear message here is that the life expectancy really rose, right? We started at 1800 and around 30 years old age, and we're now in 2012 at around an average for most of the world, except for Africa, is around 80 years old, right? And so we're living much, much longer. We also see an interesting trend that we're not going to discuss right now, but interesting drop in total number of children. But of course, that has to do also with mortality of that young age changing. And so people are living longer. And I put it in, what is the problem? The problem is brain diseases, right? So this is not exactly a problem that we're living longer. This is a fantastic thing. It just says that there are newer things that come with aging that we are seeing along the way. And one of them is our brain that is living for much, much longer than it did before. And so now we're often seeing things like fast deterioration in brain function that comes with aging. And there are things that we know a little more about, like degenerative brain diseases, so things like Alzheimer's disease and Parkinson's disease and dementia, but we don't know a lot about the mechanism. And there are things that we really don't understand, and that's the natural aging of the brain. And the process of the brain changing with age is really not something that we understand very well. And that's something that we wanted to ask here. Now, with that comes an interesting question of inter-individual variation. When you think about a group of people, and there's inter-individual variation to begin with, and when those people are exposed to all sorts of different life events, you can see a lot of variation in the way that they're responding to that. And that's something that my lab has been studying for the last 20 years. So we're looking at the neurobiology of plasticity, but from the question of who's vulnerable and who's resilient to those. So what are the mechanisms that explain to us vulnerability? For many years, we were studying the question from vulnerability. In more recent years, we started asking about resilience as well, understanding that resilience is not a passive mechanism. It's not a lack of a response, but it's actually a response of recovery. And so we're looking at what the brain looks like after stress and seeing that there's inter-individual variation. Some individuals will do great, some will get anxiety or depression or post-traumatic stress disorder. Can we find mechanisms for that? We're looking at traumatic brain injury and asking ourselves, some people have one trajectory of response and another, what is there? And in aging, there's really a big gap. It sort of opens up to a lot of variability in the aging process and what we see as cognitive function. So even if we just choose cognitive function as the matrix that we're looking at, we're going to see a lot of different trajectories there, but we don't understand really why. And we had this idea, we were going to try and ask this question from looking at it from two different angles. One is a young brain that gets injuries or insults to the brain. And we know that with injuries, with things like traumatic brain injury, even with things like concussion, there is sort of a faster deterioration of the brain. We see early dementia, we see early Parkinson's disease, Alzheimer-like, chronic traumatic encephalopathy more recently that we know of. So there's sort of a trajectory of a faster aging that we might see after injury, and put that in context together with the aging process and see if we can find things in there. Now, the problem there is very clear. The medical need is completely clear to anybody who listens to that, right? There is no effective treatments for those things. There is no disease modifying strategy. If anything, anything that's happening is trying to work on symptoms, but even not the important symptoms. We know that for Alzheimer, for example, there's a lot of funding, billions of dollars that went into drug development, but those drugs are all focused on the amyloid pathology, which we don't think is actually the most important thing there and comes very late in the disease process. And there's no personalized approach. So you might think about it as we probably all know cancer patients, and we all know the huge development that happened in cancer field when it started to be personalized, right? When we could actually follow individual patients and say, this is, this tumor looks different than this tumor, and how do we treat the patients differently? And those were the goals in mind when we approached this project, was can we identify early triggers of the pathology, understand the mechanism, identify at-risk patients, so stratify patients in some way, and then halt the progression of the underlying pathology. That's where we started with that. And for that, to get at that, you have to have a better understanding of the mechanisms that underlie this delayed post-traumatic brain injury complications and aging. To be able to actually identify the at-risk patients, you had to develop mechanism-based, quantitative, clinically applicable, predictive, diagnostic biomarkers, some kind of a pharmacodynamic biomarker, so that we could choose the right type of patients. And then if we were even lucky enough to had gone all through that, then develop and test target-specific treatments, meaning now that we understand the mechanism, can we design a treatment that acts exactly on that mechanism? So we went around that with the question of, can we actually find conserved pathways that control neural activity and plasticity to enact steps progression from healthy brain to an aged brain, or from a healthy brain to an injured brain? And I want to take a minute sort of to the side and tell you about how it all started and how we actually got to study that, because it's an interesting story, and it actually something that I think typifies the scientific process in a lot of different ways, and that you start with something in a design goal and you know where you want to go, and then along the way the project really takes you to different places. And that was one of those stories, is that we had an idea in mind, but along the way the project itself sent us onto a very, very different track and became very interesting. It started when I was a graduate student in the Hebrew University in Jerusalem, and Alon Friedman, the friend who I said I'm collaborating with all those years, he at the time was a physician in the Israeli Defense Force, in IDF, in Tzahal. And this is early 90s, right? So I was just out of the army, Alon was still in the army. One of the things that just happened is the end of the first Gulf War. And there was a mystery. There was a very interesting scientific or medical mystery, if you will, about people starting to come back from the Gulf War and showing what was then termed the Gulf War syndrome. They were showing a lot of neurological symptoms. Now, there wasn't a good explanation for that. You would think probably nerve gases and so on, but they weren't exposed to nerve gases. They actually got a prophylactic drug in the thought that they might be exposed to nerve gases later on, but that never happened. And so they were taking those drugs. Those drugs were particularly designed such that they work on the peripheral enzymes, but not cross and get into the brain. Yet those people came with signs of neurological symptoms. And so we had this crazy idea that maybe those drugs actually got access into the brain, even though they weren't supposed to. And it turned out to be true. And it led into this whole career in finding alternative mechanism for neurodegeneration that I'll tell you about now. So the first thing that I need to talk about is just explain in a minute, what is that? What is that thing that doesn't let a drug, for example, get into the brain? It is called the blood-brain barrier. Blood-brain barrier is a net of, so the brain is very heavily vascularized. There's a lot of blood vessels that get into the brain. They get very close to every cell almost in the brain, but the walls of those blood vessels look a little bit different. There's a protection layer there. There are cells and filtration mechanisms that exist there that don't exist in other blood vessels in our body, and they protect the brain. They create what we call a privileged environment, an environment in which toxins don't just get access to the brain, cells don't get access to the brain, and so on. So most of the time our brain is protected. In a healthy, young, great brain, this is what we're going to see, a very good blood-brain barrier that acts very well. But we had this idea that maybe under the stress of the Gulf War, for example, or the heat that they were in and all sorts of different physiological conditions, the blood-brain barrier was more dynamic, was breached, and now was more permeable, and the drug got into the brain. And it turned out to be true. That was the case. Published that story, and we went on to our different separate careers and starting our own labs, but we stayed with that story, and actually this developed more and more and more to start to understand what happens downstream of that blood-brain barrier. So when we started, I said the first thing that one might need is a quantitative clinical applicable way to actually have a biomarker to look at patients and say, how are they different from one another? And what we would want in this case is to have a very good way to take a patient and say whether the blood-brain barrier is breached or not. And actually, this is not just quantifying the extent of blood-brain barrier permeability, but the location of blood-brain barrier permeability. And Alon developed this MRI technique. It's called a dynamic contrast enhanced MRI. It uses things that are used in clinic, but in a different algorithm and different way of collecting the data and analyzing it. A tracer is injected into a patient, and then you are following that with late sequences of MRI acquisition. And then you can follow that data that you collected and create permeability maps in the brain. So what you're seeing here at the end of that is this is a brain that has blood-brain barrier dysfunction in some of its areas. Where it's red, it says a lot, and where it's blue, there's little, and where it's gray, there is no blood-brain barrier dysfunction at all. So we're able to follow that and quantify the distinct location and extent of blood-brain barrier permeability. So we have now a biomarker or a tool to try to use that as a biomarker and ask questions about blood-brain barrier function in human brains in this case, and then we adapted it to rodents. So you'll see pictures like that throughout the talk in people and in mice and rats. Now, why is there even a thought about why is the blood-brain barrier something that would be interesting to how I started this story? Well, in traumatic brain injury, in most of the brain insults actually, and in many diseases of aging like hypertension and diabetes, there is injury to the blood vessels. And that's something that was known, that if there's a big injury, one might see that. So in this case, you would expect that there might be some kind of a blood-brain barrier injury to that. So we started with that. We started with football players. This is an immature group playing American football in Beersheba, actually. A group of students that have an amateur league, and they're playing football there. And we were involved in looking at their blood-brain barrier at the end of the season and then a year later. And the controls were other students that were on the track team. And you can see that in a healthy young individual in their 20s, most of them showed nothing at all. So you inject the tracer, you look at the brain, there's no tracer in the brain, and you know that the blood-brain barrier worked well in filtering this out. But in the football players, you found a lot of them that had some leakage, some permeability of their blood-brain barrier. In fact, we found it in about a third of those. And those were not people with very traumatic brain injuries that were recorded throughout the season. Those were concussion or subconcussion injuries, meaning it's just them playing the season as they did. About a third of them had permeability at the end of that. And when we went back to them a year later, about a half of that third still had some dysfunction of blood-brain barrier. Now you can look at a lot of different literature and our own data for that. And at this point, we have now hundreds of patients that we looked at and that Alon scanned in Canada and in Israel. And we know that it's involved in different conditions with extensive traumatic, with extensive temporal lobe epilepsies. There's blood-brain barrier dysfunction. With early Alzheimer's disease, there's some blood-brain barrier dysfunction, and later in the disease there is more of that. And even in some psychiatric conditions, in this case you're looking at a patient with bipolar disorder that shows blood-brain barrier dysfunction. The extent is interesting, and then the locations are interesting, and it's not always the same place, but that's something that seems to come at different neurological disorders. So here came our hypothesis, right? We know now that there's a lot of different cases, trauma, stroke, aging, infection. We don't know about aging yet, that came later, but we know that injuries, there's blood-brain barrier dysfunction, and we know that there's delayed pathologies in those, things that seem common to them, cognitive impairment and neurodegeneration, and epilepsy. And our hypothesis was that maybe blood-brain barrier disruption is not just an epiphenomena. It's not just something that happened to happen in those different scenarios, but maybe it's actually involved in the pathogenesis process, meaning this is part of what drives the pathology to develop later on. So you have a blood-brain barrier dysfunction in a subset of patients that have trauma, in a subset of patients that have stroke, in a subset of patients that have infection or cancerous tumor in the brain. And maybe all of those things in the subpopulation that have blood-brain barrier disruption, this disruption by itself plays a role in the development of this later neuropathology, like cognitive impairment, or epilepsy. And if that's the case, can we find the underlying mechanism for that? Now, to look at that, we ask the questions in a lot of different ways. And you just start with asking, is it actually something that is correlated, right? Can we look at those individual variability that I talked about? So for example, this is in a concussion rodent model where we use very, very mild traumatic brain injury that repeats for 4 days. So this would be like a sports season, like those football players that we just looked at. And when we do that, we can run statistical analysis on clustering, and we find that the animals fall into two groups, sensitive and resilient. If we look at aged animals, and we just let them age naturally, we actually did nothing but wait. Those mice live to about 2 years of age. And when they come closer to their end of life, around 21, 22 month of age, and you start looking at their neural activity, you find that they also fall into two groups, resilient and sensitive. And if you are looking at those sensitive groups across the different scenarios in traumatic brain injury, in aging, we find that they actually share those two things that we thought about. One is they share blood-brain barrier dysfunction. Those animals are the animals that have blood-brain barrier dysfunction. And secondly, they share something in their neural activity pattern that is very interesting, and that's an imbalance of excitation to inhibition, meaning we're seeing a lot more activity, neural activity. Now we know that when that goes all the way to the end, that's actually epilepsy. This is what epilepsy seizures are like. Now we don't necessarily see epileptic seizures in all of those animals, but we see markers of epilepsy-like events. And then came the question, all right, so we know those two things are there. Can we now start understanding the mechanism? And that was the next sort of 15 years of looking at it. So I'm not going to give you all of it, but I'll just tell you the bottom line, the end of it is the story ended up being like this. When there's blood-brain barrier dysfunction, there's extravasation of serum protein, of blood proteins into the brain, meaning blood proteins that the brain don't see any other time because the blood-brain barrier is there, all of a sudden now get to leak into the brain. Now, this is not hemorrhaging. This is not blood rushing into the brain. This is just a change in the filtration system and in the selectivity that allows proteins that before were not able to get access into the brain to get access into the brain. And we looked at what might those be. And there are different ones that do that, but it turns out that we can pinpoint down to one protein that recapitulates that whole response. And that protein was albumin. It's a very, very common protein in the blood. I have to say, a bit of disappointing moment in my career. I was really hoping for some very rare and exotic protein that we're going to find, and it wasn't albumin that I was expecting. But that was one of those moments, you know, the data is what the data is, and it was albumin, and turned out to take us in a very, very interesting route. So this protein now gets into the brain, and it happens to bind to a receptor that exists in a part of the brain, in some cells in the brain, that's called astrocytes. And that by itself was interesting, because if you think of neuropathology, you think of neurons, right? It's in the name, neuropathology. We're thinking about neurons, we're thinking about cells that communicate with one another, and all of a sudden it was in another cell. But that cell happens to be the one that picks up albumin. It gets activated. There's this binding to the receptor activates a response, a profile in the cell that changes it dramatically and activates a very, very massive inflammatory response. And the brain knows how to do inflammation, and it needs to know how to do inflammation because our immune system is restricted to the periphery. In the same way that the blood-brain barrier blocks a lot of different proteins, it also blocks the immune cells. And so immune cells can't just rush into the brain, and the brain knows how to activate an immune response. But in this case, it is activating this very massive inflammation with no real reason, right? There is no actual pathogen to fight. And now this sets in motion this huge pathological signaling cascade, a lot of changes that happen in that cell and then change the environment of the other cells. And there's plasticity around and changes in neural pathway that finally lead to neural dysfunction and cognitive impairment. And along the years, we were able to follow a lot of the steps and understand exactly what is going on. And so this is what it looks like when we're looking in the brain. This is this red marker here depicts an astrocyte, and this green is this protein from the blood that is not supposed to be in the brain, but all of a sudden it gets taken into the brain. It massively activates this inflammatory response, which we can look at the whole genome. This is a transcriptome analysis. So we're looking across all the genes, and all the genes of inflammation turn bright red, meaning they're very, very highly regulated. When we're following this, we can say, what are the initial steps? There's changes in gene transcription, and then there's network organization, and there's a lot of things that increase excitatory transmission and a lot of things that inhibit the inhibitory transmission. So we're starting to get this excitation to inhibition imbalance that we talked about, and we can record those neurons, and after a few weeks or a month, they start showing hyperexcitability. And in some of the animals, that will go all the way to develop to spontaneous seizures and to epilepsy. Turns out when we're looking at those groups that are vulnerable versus the groups that are resilient, is that the level of blood-brain barrier dysfunction in the initial time, if you look at that right after the injury, the first 48 hours, that predicts which of the animals are going to be resilient or sensitive later on. So it does seem that it plays a role in that, or at least it's very important, and it correlates together. What happens in the later stage, when we're looking at the chronic stage, we went to naturalistic models of chronic epilepsy, like epileptic dogs, epileptic sea lions. A very interesting story about here in the San Francisco Bay in the Marine Mammal Hospital in Sausalito that we can talk about later if you want to ask questions about that. And we got some human brain tissue from patients that undergone surgery for their epilepsy, for their intractable epilepsies. And in all of those cases, we find that there's blood-brain barrier dysfunction that is chronic, and we also find activation of that receptor, this TGF-beta receptor that we talked about. So there's co-localization of this albumin TGF-beta receptor in astrocytes with where we can see blood-brain barrier dysfunction in their brain. Right. So we now have a scenario in our head to explain a lot of those steps along the way. We now wanted to see whether this is true for aging as well. As I said, we started with this idea that this might be something that's similar. And so we looked at what happens to blood-brain barrier with very natural aging. Actually, in fact, we took out every patient that had any neurological or psychiatric symptom. So those are very healthy people from the ages of 20 to 76 years old. And as you can see, In young brains, most of the individuals don't show any blood-brain barrier leakage. And this sort of rises with age, and it starts pretty early. You can see already a doubling of the amount of people that show blood-brain barrier dysfunction around the age of 40. And when you get to around the age of 70, you'd have about 60% of the people that show some dysfunction of the blood-brain barrier, healthy ones. Then we looked at the mouse brain. And what we're seeing here is we're looking at a mouse brain where all the astrocytes, all those cells that take up this protein are green, and all the blood proteins are going to be red. And in a young mouse, 3-month-old mouse, we hardly see any red. You go into a middle-aged one, 7-month-old, and you see a little bit of red, 12-month-old, later middle age, and there's a lot more blood proteins in that part of the brain. This is a part of the brain called the hippocampus that is very well known to be involved in learning and memory. And in an 18-month-old and a 21-month-old, the brain or that hippocampus is very full with the blood protein now that co-localizes with those astrocytes. And in fact, when we put side by side mice brain, and those are post-mortem human brains from individuals between the age of 20 and 70, because they're post-mortem, we could do the same type of analysis on them that we do on the mouse brain. And we could see that there's an increase with age of the amount of blood protein that we find. There's an increase in the astrocytic localization that tells us about an inflammatory response. We see a very massive inflammation signaling both in the mouse brain and in the human brain. And finally, when we move to look at gene expression, this transcriptional response that I talked about, we see that they're very, very similar in the mouse brain and in the human brain. And it's such that does predict this excitation and inhibition imbalance. This excitation and inhibition imbalance is something that we should be able to record. And so we took those tiny little transmitters and we implant them into mice, and we record them over time and see whether we can find all sorts of abnormalities. In a regular aging mouse, we didn't find any spontaneous seizures. So they don't just develop epilepsy spontaneously, but what we do see is that there's an increase in transient slow wave episodes. And the best way that I've heard anybody give it an analogy for that was a reporter from the BBC that was interviewing me and told me, this sounds exactly like this wheel of death that you get in your Mac when it slows down a little, right? And it's sort of going there, and you know that the computer is still working, but it's sort of not responsive and it's very slow. And that's exactly what this is. Those are sort of moments that the brain goes into this very synchronous, excited activity and then comes out of that. And they could be very short, they could be seconds. But when we look at the animals, we find that, again, we have this very beautiful resilient and non-resilient. So this is a young animal, and they hardly have any of those events. And we see that when we look at those, actually, when they have those events, are in sleep. So that makes sense. Then we can find a very resilient group of aged animals that don't show a lot of those events. It's a little more than the young, but not that much more. And again, mostly in sleep. And then there's a pathological group, and that pathological group have thousands of those events, of those moments per day that we find in them. And they're across the day. So they disrupt, probably you have a disruption of a sleep pattern as well in those animals. And if you take those animals and give them something that will push them towards epilepsy, they actually are very vulnerable to develop seizures. So that is something that tells us about correlation, right? We started with a question of, is it involved in the pathogenesis? And that doesn't say that yet. That says that very nicely everything fits together. We have a beautiful just so story if we were to stay here, because everything fits, and I'm describing everything that looks very similar, but I still don't know that the fact that the blood-brain barrier is disrupted is what causes this neuropathology downstream. I know that it activates a lot of signaling that makes sense, but you don't know yet that that causes that. So how do you do that? In biology, the best way to ask those questions is get in with manipulation and ask gain of function and loss of function type of approaches. What does that mean? I mean, if I want to be sure that this is responsible for that, I'm going to take a young, healthy mouse, and I will be the one manipulating the activation or blood-brain barrier disruption in their brain. And if I'm correct, and this is involved in the pathogenesis of this should be enough to get now the phenotype of an old mouse. or an old brain just from doing that. So this was one thing that we're going to do. The second one is say, so is this required? Is this sufficient, right? Is this sufficient? Is this one manipulation sufficient to phenocopy an aged brain phenotype? The other one is going to be, is it required? If I'm going to take an animal that is aging and I block this pathway, am I blocking the aging process of its brain? So is it sufficient? Is it required for that? And what became really, really exciting here is that we started with the question of, can we actually block? If we get early enough, if we go right after the injury, or if we go very early on midlife and give this and block the response, is it going to age differently? And the answer was yes. But then as a control for that, we became, we wanted to see what happens when the damage is already there. What if we took a very aged animal and then we block it? And that was the big, big surprise. And the big surprise made us extremely optimistic about the power that may lie in this. Because when we take a very old mouse and we block this, we actually saw reversal of the brain aging. And that sort of changes the way that we actually understand the process. Because the way that we were thinking about the process is very much neurodegenerative focused, right? We have cells, they're dying, there's circuits, they're not working. When there's damage, there's damage. But in this case, it turned out that this fog of inflammation that I described before that happens here, this fog of inflammation is making the brain act as a very aged brain. But when you remove that, the brain had all the capacity to act as a young brain. So I'll show you that. Now, how do we get rid of that function? What do we do with that? So there are two ways to do that. One is a genetic way. We could actually come in and sort of cut the gene out of astrocytes and create animals that do not have now this gene in astrocytes. And we could do this in an inducible way, meaning we can come in at any point that we want in the life of the animal and take it out. And we created those transgenic animals and we studied them. And another would be, can we pharmacologically block that? Can we have some kind of a drug-like property that would come in and block that? And we had a small molecule that was interesting. And there's actually, this is a person from Palo Alto, Barry Hart. He's a medicinal chemist. He read some of our early papers on epilepsy and came to me and said, I think I have a drug that you should try. And we started from there. So let's start with the gain of function. Let's start with this very young mouse brain and see what happens when we put in a pump, and this pump pumps this blood protein into the brain. So we don't even have a blood-brain barrier dysfunction in this case. We're just taking the one protein that we think is important and we're pumping it into the brain for 7 days. After 7 days, we see that we're copying the molecular phenotype of an aged brain. We get the inflammation, we get the gene transcription changes that you would see in an old brain. Then we record those animals and we see what happens with those events, those paroxysmal slow wave events that I was describing, those slowing of the brain. And when we look at that, we see that we actually get increase in them. So we also phenocopy those slow moments. What about the seizure vulnerability? We said that those aged brains were very vulnerable to seizures that we were inducing, and we phenocopy exactly the same thing. They're very vulnerable to seizure progression, and they're vulnerable to mortality by seizures in the same way that the aged animals are, those very young animals. Well, finally, we asked about cognition. Can we test cognition? And so we looked at something called the Morris water maze. There's a maze that they need to find a platform in the water. And this is what it looks like in the control animals. The control animals are animals that have the same pump in their brain, but is only receiving saline in that pump as an inert compound in there. There is nothing that actually acts on the brain. And when we do that, those animals learn very beautifully by day 9 where the platform is and how to find it in the water. But our young animals that received albumin in that pump, they received the serum protein, the blood protein, they don't learn up until day 9. Whatever we do, they don't really learn where that is and they can't find their way in that maze. Okay, so that answers all of those All of those things that we looked at, those are the 5 different things that we look at in an aged brain. And taking a young brain and infusing it with albumin, you can phenocopy those 5 things. And this mouse now, this very young mouse, after 1 week of infusion, looks like an old mouse. So let's look at the opposite of that. The opposite of that was creating those transgenic animals where we could actually take out the gene. And when we take out the gene, we could reverse the aging phenotype in young animals that receive the pump, in middle-aged animals, early aging, but most incredibly in the late aging animals as well. As I said, this idea that you can reverse it is really, it was just mind-blowing. And that was the first time, that was the first time that we've ever seen that happen. When you look at their cognition, we see that both the early aging and the late aging group, once we take out that gene once we knock out that gene out of astrocytes, now become learners that are very similar to young learners. So you even reverse their cognitive abilities. And then came this interesting point in time where we thought, all right, what do we do with that? This seems like something you really want to take out of the lab and translate out into the world. At this point, I've been treating animals that have epilepsy and traumatic brain injury for so long and decided really this is something that needs to get out. And I happen to have heard about the Baker Fellowship. I think this was the 2nd year that the fellowship was started. And they said, if you're a faculty member that have any ideas about something that you found in the lab and you might want to translate into the world, come talk to us. And so I did. And we got this fellowship that really changed the course of where my lab was going in the next 5 years, that ended up with, in 2020, a company that was started on that and will now actually try to bring that to actual patients. What we did in those 5 years is understand a lot more about drug development and start to get at a small molecule inhibitor that has drug-like properties that could do that. And here are our first results with this drug. And so you're looking here at the gene expression, for example, that we looked at. We know what the old looks like. It's the red bars that you're seeing here. We know what the young looks like. This is this dashed line. And with the drug, within 48 hours, you start seeing gene expression that looks like what it should look like in a young brain. When we're studying the molecular phenotypes, this inflammatory and gliosis phenotypes, we see that the drug brings it back to the level of a young animal. So on molecular levels, we're normalizing those aged brains. What happened with the network dysfunction? This interesting experiment actually was run like a clinical experiment. We first recorded those animals. We found those in the red group here, which are the pathological ones, the ones that show a lot of different events per day. As you see, thousands of events per day. And then we start treating them, and you actually see the reversal, and a very, very quick one where the animals on the drug become completely indistinguishable from the regular olds or the young ones. Vulnerability to induce seizure, same thing. The mortality by seizure, same thing. And most interestingly, the cognitive function. In this case, we're testing them by a different maze called a T-maze and a novel object task where we're seeing how long they remember a novel object. And the drug brings them both to the level of a young brain. So we're able, by giving them a drug, to actually remove this inflammatory fog from the brain and look at what does it look like in a brain, aged brain, with all the different changes that one might see in aged brain, but without the inflammatory fog. The other thing that the brain, that this drug is doing that we're not showing here, that it's basically, it reverses the blood-brain barrier damage. So it closes up the blood-brain barrier and creates a healthy environment, again, for the brain to be affected in. So we started with the idea that we wanted a mechanism and a personalized detection system to be able to block and even reverse pathology. And that's what we got to. The interesting part in here is this is a very novel mechanism to think about brain function, because rather than thinking about the brain cells themselves and how they communicate, we're seeing that it's the blood vessels that create the environment in which the brain cells communicate in a pathological way or not. So this vascular dysfunction, it's a novel mechanism to think about neural pathology in injury and in aging, and more data that I haven't shown in Alzheimer's disease and neurodegeneration as well. It starts in middle age, and it is associated with the individual variation and the very surprising and promising reversal potential that we found that has to do with this reversal ability to get a young brain again once it's removed. The last minute that I want to take is just, I usually get a question about, so what can I do with that? Or where can I get the drug? Or so we can't get the drug right now. It's not quite there for clinical use, but we are hoping to get to that at some point with a startup company that will develop that. But what are some things that people can do? What are some things that were shown to be involved in this vascular health? So some things you can't do anything about. It's predisposition. For example, the one gene that is most known to be associated with the risk for Alzheimer's disease is ApoE. And there's a polymorphism of ApoE that's called ApoE4. When people have the ApoE4 variant of that, they're much more susceptible to develop dementia and Alzheimer's, and that turns out to be something that causes a leaky blood-brain barrier. But there are other things that are involved in vascular health that we know from the periphery, things like blood pressure and like diabetes, and both of them are involved in the health of endothelial brain cells and brain vasculature and microvasculature. And so taking care of controlling things like blood pressure and glucose levels are very important. And then lifestyle changes like sleep and stress and physical exercise seems to be involved in that. We don't know a lot about mechanisms yet, but those are things that will come in the next few years, I think, to look at. But definitely things that can help vasculature health. So I'll end with that and open it up for questions. And thank you guys for listening.
Amazing. Thank you so much. I wanted to remind everyone to put questions in the chat or send us a note that you want to ask a question so we can call on you. And while you're doing that, I thought I would start with a first question, maybe to step back and just ask you about the picture of collaboration and how that works in a long-term study like this. How did you collaborate with your partner, Alon Friedman? How does it work to work between labs and also to shift between clinical studies that look at rats versus looking at people? You even mentioned the marine mammal center here. Sort of how does it work to do research on different groups, but also in collaboration with others, the amateur football players in Israel, if you've done studies with groups here in California, just sort of a picture of how it looks to do that kind of collaborative work.
Yeah, so the collaboration with Alon is that was one of the most fantastic. This is like the ideal collaboration that anybody I think can hope for. We're very good friends for all of those years, and we bring very different expertise. Alon is well, not a clinician anymore, but was trained as a clinician, as a neurosurgeon, and then switched to do full-time research, but from a clinical perspective and working with people. And we were giving the side of thinking molecularly, thinking about mechanisms, thinking of what can we add to do in animals. And it's been one of those fantastic things, not just for us, but it really percolates down to the whole group. And so students from Alon's lab will come and spend time in my lab. Student from my lab will go to Ben Gurion and spend time there. A lot of them people that would have never gotten to Israel in any other way. So it was nice to expose that to them. They always come back, big supporters. And we have a very active collaboration that we try to keep this way, not just between the two of us. We probably talk a couple of times a week, but with the students. For example, this morning we had a joint journal club and lab meeting with the group and now we have to find times that work for Berkeley, Beersheba, and Halifax. with a few people from Bonn that were joining in. And so we have that. Next week, we have 2 days of a lab retreat where all of the people from my lab and from his lab are going to present so that we all know each other. In different times, we used to actually fly to Canada, and people from Israel would fly to Canada, and we all would meet there and have it in person. Clearly that was better, but we do what we can. And from that also came, I think, the very joint thing of we're looking at people, we're looking at animals, we're trying to get what we can from the animals, and then with those understandings, go back to patients and see, can we do things? And indeed, it's now bigger than that. And so we have actually a collaboration within Berkeley and with the Aging and Memory Center and with the VA at San Francisco, looking at different aspects of patients as well. And in asking in all of the different projects that we're doing, we're always trying to go now back and forth between what can we learn from the rodents and how can we translate that to patient. And, you know, that's at the end, that's the goal, right? It's not to help rodents, but it's to help patients.
I wanted to understand, and maybe you said this at a more technical level, but what makes some sensitive and some resilient from sort of the very beginning in young or healthy rats or young or healthy people?
Yeah, so you know, there's always a question of the chicken and the egg and going back and peeling it down. And so we have now biomarkers. We know, for example, in people with the traumatic brain injury or with the animals of traumatic brain injury, we know that the ones that show early blood-brain barrier dysfunction are ones that show sensitivity. In the stress story that I haven't talked about today, I just mentioned in the beginning, it has to do with myelin and changes in myelin that we know are correlated with sensitivity to, they follow in different brain areas that show you different ways that those patients and animals then present. So some of them present avoidance behavior if it's in one brain area, and other presents fear behavior learning in another brain area, and so on. So you can get a lot of that, but then the next question is, well, why do some people have blood-brain barrier deterioration in agings and others don't? So that's the question, one of the basic questions that we're now tackling in the lab, is trying to then take this stage and say, why do we see blood-brain barrier dysfunction in some individuals or not others? Why do we see myelin changes in some individuals and not others? With the myelin story, for example, one of the most important lessons that I've learned there is that when you look at the resilient animals, they've had the biggest change. And so when we looked at all the different pathologies that might occur, the different domain, acute threat reactivity and so on in the groups of them, and then you had the group that didn't show any differences in behavior. They actually seemed protected, but when you look at their brain, they had the biggest change in gene expression. And so there was a very active mechanism there to confer resilience, right, and recovery from the trauma. And it might very well be that in aging, it's not necessarily that in some people there's sensitivity because things happen, but maybe in patients where we don't see that, the very healthy agers, the center in San Francisco, the Aging and Memory Center, they have a group that they call super agers. There's people in their 90s that don't show any deterioration, and in fact, some of them show improvement of cognitive function every year that they come in. So what happens in those people, right? There is probably something going on to confer that resilience as well, and we know very little about that still.
Great. I think this is a great place to conclude, and I want to thank everyone for joining, and thank you so much, Daniela, for joining us today.
Thank you for the opportunity.
Thank you all.